meeting or the webinar. Friends, just to give the formal information to our two speakers, eminent speakers, who have really contributed a lot. And I saw the pamphlet where very remarkable work has been done by these distinguished scientists. For them, the Indian Council of Agriculture Research, National Dairy Research Institute, NDRI, the canal plays a very crucial role in developing quality human resources for the dairy sector of the country by developing need-based educational program and introducing innovative in learning style of pedagogy. We have a very nice setup in terms of the dairy production, dairy processing, and dairy marketing. In dairy production, we have the other very good setup, Animal Biotechnology Center, and where we are working on the biotechnological aspects in order to enhance the productivity from the animals. Here, the title which has been given, this is on the genomic, this harnessing the potential of genome editing tools to augment the productivity and health of the farm animals is the need of the hours, particularly for India, where we have a large number of less productive animals. And there is a two-way, everybody knows that in order to have multiply these animals, there is one way is just take the conventional genetic breeding program, and which will take more time. Everybody knows that. But the talk related with this seminar, where we can go for the gene editing, and we can multiply the quality animals at the faster rate. So therefore, the title which has been selected by Dr. Naresh Shilokar and his team, Dr. Manoj Kumar Singh, and also taking together with the very eminent people, very eminent researcher, like today is Dr. Jialong Wang and Dr. Mike McGrath, is going to enrich our knowledge, the methodologies which can be useful to us, and many more things we will learn from them. And these two lectures will enhance our knowledge and also the further implementation of that knowledge in terms of producing the genetic edited or gene edited animals, which is going to give us the maximum advantage in terms of the production, productivity, and many more. Thank you very much for joining us. This webinar, and I once again welcome you, and thanks to these eminent speakers for sparing valuable time and joining and willing to wholeheartedly willing to deliver the lectures here. Once again, I thank you, and now let us proceed further. Dr. Naresh.
Thank you, sir. Thank you for your always motivating us to do the good science as well as contribute to the science. Thank you once again to you and the Honorable Joint Director, both the research and the academics and the head of the department. Now we will proceed to the presentation. First were presentations from the Wang Xianglong. He is currently the professor at the Northwest AF University, China, and he co-leads both the animal model for development of molecular genetics research efforts dedicated to the improvement of the small ruminant animals, particularly sheep and goats. His lab is mainly applying the genome editing tools for the genetic improvement that ultimately promotes the sustainability, animal health, and the rapid increasing the meat and fiber production, particularly small ruminant sheep and goat. Uh, he has been produced these genome-edited animals, particularly for the improved fiber of the Kashmir goats as a model to understand hair follicle biology, to use the indigenous sheep breeds to test the variation associated with heavy muscling as well as wool production and adaptation to the climate change and parasitic resistance. Before he has been completed his PhD from the animal breeding from the Technical University of Munich, Germany. Now I invite the Dr. Wang for his the expert talk. The Dr. Wang, please.
Okay, so thank you for your introduction. Can you hear me right now? Can you guys hear? Okay,
Yes,
great.
yes.
Then I will start. Okay, so I'm really happy to be invited here for this talk to the Indian community, because I know that the Indian has also have a large number of the small ruminates, which I will talk about the sheep and goat genome editing for genetic improvement. Okay, so before I start my talk, we will have a quick overview of the development of genetic reproduction physiology that lead to genome editing. And after the animal domestication, people actually have made many efforts for especially for the artificial selection and also for crossbreeding in the last 200 years to record the statistics and also the phenotypes for genetic selection. And the people also made some reproductive efforts to help, like to help with the embryo transfer, nuclear transfer, and also the artificial insemination, and also this sperm sexing. And in the last 20 years, we came across with the genomic area with the help of the quantitative traits loci, and also help with the marker assisted selection, and then followed up with the many omics-based technologies, including the proteomics, epigenetics, microbiotics, and then another approach is to using the genome editing approaches to quickly generate the animals with desirable traits in a very short time. So here I quick reviewed the development of the livestock genetics in the last 200 years, and then come up with the new technology with the emergence of the site-specific endonucleases, which people are recently using, especially when they came across the emergence of the CRISPR-based technology. And the CRISPR-based technology has many merits. I think most of you will know that, which is simple and which is first more efficient than the previously used methods. And this is also time-saving and affordable, and also is more process and more rapid, and we can also easily to make the multiplex editing. So here, as the CRISPR-based technology also have many usage or applications from biomedicine to agriculture. The people are here all know that in the last year, there are two Nobel Prize laureates, these two ladies, meaning that the CRISPR technology can have the very promising applications in the future, not only in biomedicine, but also in our area, which is agriculture. And with the CRISPR-based technology, and the people now can easily integrate it this technology to either from gene disruption or to some specific single variant replacement. Here is some figures that indicate the illustration of the mechanism that CRISPR-based technology, including the Cas9 base editors and also the recently emerged prime editors they can do. And but here why I will work only in sheep and goats, not in other animals, because sheep and goats are the first domesticated livestock species with a long history, and also in China, the sheep and goats has the largest populations over the world, both in the number of the sheep and goats. We have the largest population over the world, and we now have the 1/3 population of the total sheep and goats in world. And also, that sheep and goats have many advantages, including their production with multiple purposes. They can provide meat, milk, fiber, skin, and also they can be considered as economically valuable fibers such as the cashmere fibers, which I know that India also have some cashmere goat breeds. And also this is an essential source for some small holders. I think they have the same similar situation also in India. As their gestation period is not that longer compared with in cattle, and they can also like provide considerable meat production levels. That's why in our lab we select sheep and goats and models for genome editing. And here is a representative example. And previously, while our student, he published 2 review papers and to revealed that how we can do using sheep or goat models. Here we can see that not only have the agriculture applications, but also can be for animal welfare, for the pharmaceutical applications, for the veterinary applications, and also for biomedical applications, like to modeling the homing diseases and also for the xenotransplantation. So here I will start with my story. Our first story, we do the genome editing in sheep. Here you can see a flock of the Chinese tan sheep, which is a Chinese domestic sheep breeds that have been raised for thousands of years in the northern part of China. And our aim is to improve the growth fat, the growth rates, the body size, and also for the litter size, and also to shorten the tails, because in these breeds, the length of the tails can be accounted for like 1/3 or sometimes 1/4, meaning that but in these fat tails, they're all like including some fat that people don't like this any longer. And our another aim is to remove the horns, because for some standard meat production sheep, they are all hornless. So therefore, our aim is to improve these five phenotypes in this tan sheep. Our first story is we first targeted the myostatin genes and also some other 3 genes. We first tried these 3 genes. The first one is myostatin. The second one is ACP, which is a gene that associated with black and white coat color in sheep, and also the BCL2 gene. This gene is associated with the fat color in sheep. And after the experiment, we totally obtained 36 live lambs, and the single gene targeting efficiency is around 30%, and for the simultaneously editing of 3 genes, the targeting efficiency is around 6. And next, we are considering whether the targeting of these 3 genes yielded desirable phenotypes. So first, we measured the birth event and also the body event for each month. We measured these phenotypes for 2 years, and here you can see from the figures, the mild-standing disrupted animals represented a higher body weight compared with the control groups, meaning these phenotypes can be used for sheep production in the future. And we also targeted the mild-standing to see whether it is possible to target with larger fragment deletions. And here we conduct this experiment again to totally obtain 35 live lambs. And also we can see from the phenotypes that's large deletions and also the single mutations yielded large body weight compared with the control groups. And from the Western blot, we can see that in the edited animals, the expression of the protein was decreased compared with the wild types. And if we see the phenotypes, we will see that for some animals, they have the very large deletions within these genes. And we also tried the mild-standing again to check whether we can get this bioallelic editing, because previously you can see that some of the animals are heterozygous animals, and we also see from the animals we see that the body event did not changed simultaneously, and even we are able to obtain the animals with the bioallelic editing, and but we can see from the HE staining figures and also the transmission electronic microscope that the mutant animals, they have the larger fiber diameters compared with the wild types. And the second gene, the BCO2, was considering with the fat colors. And here we can see that from the phenotypes, the animals with bioallelic disruption, they indicated with some a little bit of the yellow color fat in their body fats. And we are considering, people are more considering that off targets yielded by the genome editing technology. Therefore, to discriminate that the variants in the gene edited animals are either inherited from their parents or are really generated by the technology, we designed such study, the trial-based whole genome sequencing, to see whether the variants in the gene edited animals are highly inherited or are generated from the technology. And we totally sequenced 3 trials, and also using some pipelines to eliminate the variants that are inherited from their parents, and also these variants existed in a very large sheep variant database. And so here we didn't show the detailed results, and I can tell you that in the gene edited animals, the variants the number of the de novo variants, which is we we determined the de novo variants, meaning these variants are de novo as they are not inherited from their parents, that are new variants after in the offspring. The number of variants are around 20, which is similar. This number is similar to the de novo mutations or de novo variants that are in any offspring in many species, like in homins. In each generation, we will generate like some no larger than 100 de novo variants in each generation. So meaning this de novo variants in the gene editing animals are not generated by the technology. So here we also like targeted another gene named SOCS2 because previously this gene was reported to be associated with the body size, and this time we used because the target site was the C2T replacement. Therefore, we use a new technology named CBE, cytosine base editing, and from the results you can see that 3 edited animals they yielded relatively larger body weight compared with the control groups, and we did not observe any off-target mutations in these 3 animals. And here we also targeted another gene named BMPR1B, which is the most famous gene in sheep for the litter size traits, because in this tan sheep breed, it is a very old Chinese sheep breed, and within this population, the genotypes of this are GG genotype, which is genotype associated with higher litter size, is very, very rare, this genotype. Therefore, we want to generate the GG genotypes for the future to yield a higher litter size. Therefore, we targeted this variant, this AG variant, using the ABE, the new technology ABE. And here you can see when we use the SSODN before, we successfully obtained the animals with the A2G replacement, but the target efficiency is very low, and we also observed a large number of the heterozygous animals. But when we use this ABE, we can see that the target efficiency is very high, and also the animals with these heterozygous and is not as large compared with the SSODN approach. And here is this study is the first report of the ABE editing in large animals. Here you can see that we actually observed some bystander mutations are indicated in blue to see that this technology still need to be optimized in the future. So here I will talk about our story in goats, and I think as also in India we have one breed named cashmere goats. They produce very high quality cashmere, and also for the goat production, we suppose that the meat, because the price of meat now is exceptionally larger than the cashmere can produce. Therefore, we want to also to improve the meat production in goats, and also we want to enhance the litter size in goats. Therefore, we will finally target the 3 phenotype aims. And first, we targeted this myostatin and FGF5 genes. These 2 genes are also the superstar gene for the meat production, and also for the fiber length. At the beginning, we were successfully generated animals with 2-gene disruption in goats, and the targeting efficiency in each gene is around 20, and in the 2 genes disruption it's around like 10%. And because this study was from 6 years ago, and later on we improved some technologies. The phenotypes in these 2 genes actually was very successful. For example, the fiber length, we want to the length of the cashmere grows much longer than the normal goats, and we measured the length of from the staple, which is the more wild one, and also the cashmere, and also the cashmere yield, cashmere diameter, simultaneously from the beginning, from after the animal birth, the 2 years. And we see all the results. Here you can see that's the gene edited animals. In gene edited animals, we observed a higher fiber length. And we similarly we also observed the higher body weight in gene edited goats using the same target gene. Here you can see we measured the body weight for many months. So this is the goat models. We measured the phenotypes. Okay, so next we are also considering that there are some off-target analysis using the whole genome sequencing. And here you can see we are not only measures the single nucleotide variations, and we also measures the indels, the scene copy number variation, and also the SVs. We use the similar technology pipelines to eliminate the variants that I either inherited or are new variants. And we also validate all the variants with the Sanger sequencing. And here you see that we only manually determined 2 indels as off-target mutations, and especially in the de novo variants in F1 animals that could be attributed to the nucleases, which is compared with hundreds of thousands or millions of the variants that can be neglectable. And here you can see we target another gene, the GDF9, which is also a gene that associated with the litter size in goats. And because of this, in this study, we use the SSODIN, previously the old technology, that targeting efficiency is not that high. Later on, we used the base editors to introduce the stop codons in FGFL to see whether there will be some similar results that can be done as we did previously use the Cas9. And here you see we totally generated 5 animals, and 4 animals with edits and 1 with the wild types, and we measured the fiber length in each generation. And here you see the phenotypes is very apparent. And here are some animals, the figures from the animals. And here is the animal studies we have done. Actually, every year we will produce like animals with 2 or 3 genes targeted with different purposes. And people are also considering the safety of these new technologies, especially the base editors. Therefore, we conducted another study to see whether the base editors, which encoding the CBE and ABE, they will induce some negative efforts in gene expression levels as also in splicing events. This study was published, I think, 3 days ago. We actually sequenced a large number of data, and we will see that the editing induced many differently expressed genes between different groups. And we tested these base editors using different variants, and we have not identified the differently expressed genes and also some splicing variants. And we further validated these variants. We randomly select some DJs and also splicing variants, and we validated it, meaning that the consideration of using these base editors, especially for the gene therapy, need more consideration and fully characterization of the negative efforts that were derived from the base editors. And for our future, we also reviewed the sheep and goat genome engineering from these two studies, while our international students from Egypt, he reviewed the progress and also the history of the genome editing, especially in small ruminants. If someone is interested, we can have a detailed discussion later on. So with the emergence of the prime editing in 2019, our group was followed up to see whether this technology, because this technology is able to induce all types of the single nucleotide variations, all TRF types. So we are considering whether this system will be applicable in animals, especially in livestock. But first, we try this in mice, because mice is easy to handle and we can easily get the new results. And here you can see from this mice study, actually we get some results with some at some target loci, but the efficiency is the targeting efficiency is very low. As you see, the maximum the frequency is around 10%. And later on, we did some optimization to see whether this this system is able to working even more better in other types. Therefore, from our recent study, we improved this method, and maximally the targeting efficiency can increase like 5 times compared with the conventional methods. And we also did some other further development in the prime editing systems to improve the targeting efficiency. For example, in this study, we improved the prime editing activity by using the poly-U in the page RNAs, and also we see the mutant PEG RNAs will be more active for prime editing. This study was recently under review. So I don't know the time. I don't remember. Maybe like half an hour already. So I will quickly make the concluding remarks. And we show here that the genome editing in livestock, especially we targeting different genes for different phenotypes in one animal, is feasible in animals, because in our study we normally can target 3 or 4 genes in one embryo type simultaneously. Seems feasible for, especially for integrate many phenotypes in one animal, which is feasible in the future. And in sheep and goats, we are not only like to improve the phenotypes, we can also like to use this technology to validate the gene functions, like to introduce the disease resistance, generate bioreactors, and also modeling human disease in the future. And we also think that the more investigations is required to evaluate the influence of especially the food animal products derived from gene editing. I don't know how the situation is in China, but I have more discussion with guys in States and also in Brazil, Australia. It seems they have also made some positive efforts in the future. We also think that the more regulatory plans are required regarding to, especially for animal breeding, to transfer these animals from lab to the farm, because in China, our experiments are restrictionally regulated by the agencies. I don't know whether in the future we can easily like to transfer these animals to the farm, and let the breeders can use these animals with very high quality phenotypes. And we also think that more studies is needed to improve the efficiency and the accuracy, especially this accuracy, because you can see from here that the targeting efficiency is acceptable, but the accuracy, like for some bystanders in base edited animals, is still need some improvement. We also think that regarding the application is we need a future and many work to be done to finally suit this usage in the farm animal or the food animal systems. So here are my concluding remarks. I thank you for my collaborators in Damascus and also from international, and also I thank you some our group members, colleagues and students, and also some fundings from national and from local. Thank you for listening. I guess I can see from here there are already some questions raised, so I hope I can answer it. Thank you.
Thank you, Professor. Please unshare your screen. Hello.
Yeah, I can.
Yeah.
Okay.
And we will go to thank you for your presentation. You have given excellent presentation, particularly that application of genome tools in sheep and goat. And I was just counting how many animals you have produced. Is more than 100 animals, that the edited live animals is produced by your laboratory. And there is an excellent achievement from your team. I think I must congratulate you for achieving the large number of the edited animals you have been produced through the application of various editing tools like base editing, prime editing, and also the genome editing, conventional genome editing. Thank you once again. I think as you mentioned is that there are a couple of questions I see in the chat box also, Q&A box, there are a couple of questions. So I will just be mentioning the questions. You please respond to them. Or if you would like to directly respond to them, you can just go ahead. So it's better to, I will ask you the questions which have been mentioned in the Q&A box, and then you respond to them. Please unshare your slides once again.
Okay, thank you. So you mean, okay, yeah.
One question, D and Das is asking, is there any negative impact on the health and physiology of edited animals with the targeted gene?
Health or physiological? Okay, very good question. The first question, the healthy and the physiology conditions. Actually, we are also like concerned about the healthy or the physiological conditions. We actually measured the blood chemistry in all the edited animals, as we did in the hospital. And for this blood chemistry, all the live animals are very well. And for the healthy conditions, we came across some scenarios that for the myostatin edited animals are too large, raising difficulties for the delivery. I don't know whether this is a healthy problem. Then later on, we take some actions like to reduce the nutrition at the end of the gestation period to not make the animals too large for the gene editing animals. And we also like to take some actions in encoding, like we transferred more embryos and to produce like at least or maximum 2 animals from one mother. Therefore, their average body weight will be normal, not to leading like difficulties in delivery. I don't know whether I answered the question.
Yes,
So meaning,
I hope you have answered the questions which is rightly to
yeah.
Ian Das. The next questions from the Dr. Vasudev Kushwa. He was asking about the how to identify gene of interest for gene editing, particularly those have the negative effect.
Okay, yeah, great question. So for the last question, if you are interested, you can check our published literature that we actually measured the blood chemistry parameters, like for at least 30 parameters in our edited animals. Okay.
So, another, yeah, thanks, Dr. Wang. One more
Okay.
from the Dr. Kushwa. When we using the multiple gene editing, why the efficiency of the gene editing decreases when we use the multiple gene editing in one go? That is multiplexing he's been talking about.
So when we use multiplex editing, what I didn't get it.
When we use the multiple gene editing, multiplexing, the editing efficiency decreases.
Yeah, when we're using the multiplex editing, the targeting efficiency for multiplex will be, for sure, will be like decreased.
Yeah, so he is asking why this is decreased efficiency.
Because yeah, this is the if you targeting one gene, the targeting efficiency is like 30%. But if you're targeting like 2 genes, it needs to like to the frequency is you need to like one frequency times to another frequency. This is for sure that the frequency will be decreased.
Oh, okay, thank you. Next questions from Ms. Kamlesh Bajwa. She's asking about the did you opt for direct sequencing for identifying mutations in the target site, or also perform the survivor assay or T7 assay before going for sequencing?
I didn't find the question. The question is in this box or?
Yeah, QA box. Yes,
Okay.
QA box. Because I'm skipping some questions because of the shortest time. I'm taking those questions which seems to be a necessary answer from here.
Oh
And
yeah, oh yeah, yeah. Okay.
Right to you,
Yeah.
then you can respond to the particular attendee.
Oh yeah, thank you. Okay, I can see that. And did you opt directly for seeing that in the mutation? Okay. 5 years ago, we normally identify the mutations in target sites using this T7E assay and also the Sanger sequencing. But now we can easily genotype the mutation with the deep sequencing because the price of deep sequencing is very cheap right now. So normally we use the Sanger sequencing first. If we have some interesting sites, we subject it to deep sequencing, to the targeted deep sequencing, meaning that the deep sequencing for a very short sequencing.
Yes, thank you. There is a right way to do the all editing experiment. Thank you. We have 2 more questions because we have the 5 more minutes. So just a few more questions. Misha Soman is asking, is the full length sequencing necessary for gene editing work?
Is the full length sequencing necessary for gene? Okay, so I think the answer is same to be the second question here, how to identify the genes of interest for gene editing, because the decision of which gene is, I think, the most important decision before the gene editing starts. You need to determine that which gene you should work at. This gene normally should have with the known functions. The functions should be very clear, as the function of this gene should not bring some lesser issues for the animals, like to bring the abortion for the animals. Meaning that the function of this gene should be very clear. Therefore, I think the sequences of this gene will also be clear, because as right now, with the perfect annotation of the sheep and goat genomes, nearly all the genes are well annotated. Therefore, the full length of sequences you can easily find. Okay.
Yeah. This is just because this is a common question that many the people they were asking, do the genome edited livestock need to be regulatory approval for the release or commercial farming, from the Rakesh Kumar. So he's asking about the regulatory issues in the genome edited animals.
So where is the question? Is the last one?
No, not last. After the Misha Suman, this is the second one.
Misha Sama. Okay, yeah.
No, Rakesh Kumar is asking about these. Do the genome edited livestock need the regulatory approval for the release and the commercial use? I think this is a yes.
Oh
answer.
yeah, okay, I see that. Yeah, for sure, yes. For sure, yes. The gene editing livestock, they have a very complex approval for their release to the commercial farming systems. Especially in China, no animals has been approved. No gene editing animals has been approved to commercial so far.
Professor Wang, because we are running up the short of the time, I will take the two important questions, one from the Sujoy K. Dhara and another from the Mike McGrew, and one more from the Dr. Satish Kumar. So from the Sujoy K. Dhara, you just quickly respond. Did you use the genome edited animals to produce the next generation animal? This is the first question. He has the three questions. So in the last, if you go in the last, he has the last questions. Second one, did you have the chance to check the genome of the newly generated animals? And was there any reversal of mutation in these animals, Those are genome edited. The last one.
Oh yeah, okay, yeah, for sure we did. We used animals to cross with the normal animals, and we also use the gene editing animals to cross with another gene editing animals. And from the genotypes to the next offspring, we observed that the genotypes was the same as the gene editing animals, meaning that the edited genotypes can be transferred to next generation.
Oh, okay. So the next question from Dr. Satish Kumar: how can we increase the biallelic mutation using the CRISPR-Cas9 approach? Please suggest any plan. Also, he has one more question. So I think Dr. Satish in the chat box. BMPR1B is a gene. It's in the small tail can have a ship, which is already a proliferant carrier for Borrelia gene, which is, will this editing will improve the litter size further in this prolific ship? Please suggest the CRISPR-Cas strategy which can target 2 or 3 genes simultaneously. So this question is some of the question in the chat box also. So can we quickly respond that one question that you have edited the BMPR1B gene in the small tail hand ship, which is already prolific and carrier of the Borrelia gene?
I didn't get your question. Sorry.
Okay.
So It's meaning that this BMPR1B gene.
Yes, yes, yes.
Yeah, I know this gene is
This gene is
derived from India,
and clear of the,
like many years ago. This gene was from Indian breeds, right?
okay. So I think I could not get more. Some more questions are there, but since we are the running of the short time, maybe
Okay.
Mike is here. He has some questions. So the Mike McGrew, you wanted to ask something directly because your question, I was seeing the question when you sent, but I could not find in the either in the chat box. Maybe it missed.
Yeah, sorry. My question was when you carry out multiplexing, what do you think the risks are for having chromosomal fusions, by doing two cuts into a single cell? Do you think that will, this is for Dr. Wang, if he thinks this will be a problem, because we all worry about having multiple cuts and causing chromosomal rearrangements.
Okay. Yeah. Great question, Mike. So we actually considered all this situation. So this fusion of the genes, if these genes are in separate chromosomes or like far away from each other, so I think that this situation wouldn't be a problem because from our experience, every year we target like 3 to 5 genes, or we injected 3 or 5 genes into one embryo, and now like this situation happened before. So I don't know whether I answered the question.
That's fine, thank you.
Okay, thank you.
Okay, thank you, Dr. Wang, for your excellent presentation and responsive questions. Few more questions are there. So what best I can try, I can write to you, and then we will send your responses to the respective attendees who has been asked the questions. Thank you once again. Please answer your slides. So next we have presentations from the talk from the Mike McGrew. Just once again, Mike, you are here?
Yes, I am.
Yeah.
Here's my video.
Thank you, Mike, for accepting our invitation to deliver the talk at this symposium, targeting, particularly highlighting the importance of genome editing tools in farm animals. So, dear attendees, Dr. Mike is currently a senior lecturer at the Roslin Institute at the University of Edinburgh, United Kingdom. Because everybody knows the University of Edinburgh, who has been pioneering in the field of the somatic cell nuclear transfer as well as genome editing. He received his PhD from the Boston University School of Medicine, USA. He carried out his postdoctoral research on the chicken embryogenesis at IBDM Merily, France Institute, and on chicken transgenesis as Roslin Institute, again postdoctoral. His laboratory currently studying the type of the stem cell in bird and primordial germ cells, which produces the sperms and the eggs. These cells can be used to generate the gene-edited chicken which can contain the precise changes for the genome. He also works on the gene editing and use the sterile surrogate chicken host to create a biobanks, frozen aviary, using the germ cells. And the avian bank will add the effort to both the manage and conserve both the rare and industrial breeds of the poultry. And he's the one of the pioneering researcher in particularly for the poultry science, highlighting the importance of the stem cell as well as the genome editing. Now I invite the Dr. Mike for his talk. Dr. Mike, please.
Thank you, Dr. Selukar. And can you see my slides now?
Yes. Your slides are clearly visible.
Good, thank you. And you can hear me. Can you see my pointer?
Yes, pointer also.
Okay, that's fine. Well, thank you. First of all, I'd like to thank Professor Chauhan for the nice introduction to your institute and to our lecture series today. And I'd like to thank yourself for organizing this lovely lecture series, and I hope it will be informative to the delegates who showed up. And I'd like to thank Dr. Wang for his lovely presentation and the nice introduction to the field. So I will just note the time for my talk, and I will try to speak for 45 minutes and then take your questions. So I thank you for the invitation. I'm going to talk about the work of my laboratory, which, as you heard, is at the Roslin Institute. And we work on precision breeding and reproductive technologies for bird species. So this is quite different than what you've been hearing about the work being done in mammals. Birds are quite different, and they have particular problems for both breeding and for conservation. Oops. There we go. There we go. Sorry, that's the next slide. So birds are an amazing class of vertebrates. There are about 10,000 different species that are scattered over the planet, and they live in very diverse environments, and environments and conditions, and they all produce offspring through hard-shelled eggs. How they adapt to their environment is very informative for us in understanding life and evolution. And it's also very important as we have been changing their environment. So it's very important to understand how they're going to adapt to the future environment caused by climate change. Birds, as you know, have a very close interaction with humans, and they've been raised for hundreds of years, especially this bird down here on the left. This is Gallus gallus. This is the chicken. This has been raised for thousands of years by mankind and has been selected to have very different body adornments, and has been raised in very different environments by man as we migrated across the planet. Okay, so for birds, as you know, chicken is probably the most important bird species, and it also is the most abundant bird species on the planet and the most abundant livestock species. So for chicken, there are over 60 billion chicken raised on the planet each year. That's about 8 chickens for each person raised per year, which is an astonishing number. This is just showing their global pattern or production pattern over the planet. You can see there's a lot raised in temperate zones across the planet, but the highest density is in China and India, and also in Europe. And several billion chicken are produced yearly in these countries. And I want to point out that a lot of poultry production is being carried out in very tropical environments. Chicken is adapted to live in all these environments, but productivity seems to be best in temperate environments where the temperature isn't too warm or it's too cold. So we're a little concerned about, with climate change changing the ambient temperature of the planet, how poultry production will adapt to these changes. Oops, sorry, I'm having trouble seeing part of my slides. Okay, so chickens can basically come in 2 different types of classes. We have the indigenous breeds of poultry, and also we have the highly selected commercial breeds of poultry. Indigenous breeds of poultry have been adapted to their local environment, and they've lived there for hundreds or thousands of years. They're very good at scavenging feeding in these local environments, and they have very high biodiversity. They also have lower productivity because they have low body mass that's put on, and they also lay fewer eggs. We think part of this might be because they are adapted to more environmental insults and also pathogens, so they put more of their energy into surviving in these environments under low food input. Whereas on the other hand, commercial poultry have been selected by man very intensely for the past 50 to 70 years. They are highly productive, and they, in their controlled environment conditions, so if they are maintained under proper conditions with correct temperature, they're highly productive and they're a very useful food source for all of us. Their productivity depends on a very high nutrient diet for this. So if they are given less food, of course, they will produce less flesh, less meat to eat, and also less eggs. Oops. Sorry, I seem to be.
Yeah.
Okay, sorry. Seems to be moving in different ways. So basically for this talk, I have two large or major questions I'd like to bring up or address. One is how we can use genome editing to apply to better both systems of poultry farming, one the indigenous breed and one indigenous breeds of chicken, and also for a commercial poultry farmer. The second is, can we develop new reproductive technologies for poultry conservation and biobanking programs? And I wanted to mention at this point, I keep talking about indigenous breeds of chicken and commercial breeds of chicken. As a point of, as an example, we can talk about India. So India now has a very developed economy, very resource and economically rich, as you know. Indigenous farming now provides about 40% of the indigenous breeds of chicken provide about 40% of the poultry farmer for India, where the other 60% comes from commercial breeds. And with time, as this goes on, the indigenous breeds are being lost and they're being replaced by commercial breeds. So the idea is, how can we use genome editing to actually improve both breeds of chicken, and also how we can use reproductive technologies to safeguard these different breeds of chicken. Okay, so I'm going to give just this basic background of genomics and genome editing, even though we've already had the beautiful introduction by Dr. Wang. So you could imagine that each of us is, each animal contains a genome, and this genome could be considered as a book. And this book contains all the words, all the chapters, and the letter that spells out what we are, or actually the genetic code to make us. And if we look close at this book, we could see that it's made up of pages, and the pages contain letters and words. And some of these letters and some of these words make for a more productive animal, and some actually are deleterious mutations that lead to misfunctioning in us and also to genetic disease. So traditional breeding relied on chance mutations that occurred in this book or in these pages of letters. And this would lead to, in this example, a simple deletion or change of one of these letters of genetic code that will make a change in the animal. So traditional breeding would be selecting for this animal that had this naturally occurring change, and if it was profitable, we could select for that animal for increased productivity. We could also add, in plants, they add chemical mutagens to increase this process. So by irradiating to the plants, we can cause random mutations, and then that plant can be bred from. This type of editing isn't regulated. It's accepted in all countries. So even though it's a bit random, this is actually accepted compared to our genome editing tools. So that brings us to genome editing. And this is why we're so excited by this tools, that instead of randomly generating mutations or identifying existing mutations that exist in natural populations of animals, we can go into this book made up of these letters, and we can simply change a single letter or delete a single letter in one word in this entire book, or we can actually rewrite it and change it to the letter we want. So changing a word in the book and changing that animal. And that's the power of genome editing techniques. As you heard from Dr. Wang, the genome editors come in 3 different flavors. They're site-directed. All of these are site-directed. Sorry. All of these target a location in the genome. They cut the DNA at that genome, and it leads to 2 different processes occurring. If you give no repair template, this leads to a process called non-homologous end joining, which will lead to a tiny indel deletion or an insertion at this location in the genome. If you give it a template, it leads to a process called homology-directed repair. And this can lead to precise genome editing. So we can go in and change a single nucleotide at a particular location, or we can actually change the whole gene at that location and take a coding sequence for one animal and introduce it into another. And this is called precision breeding or precision agriculture. So we're investigating the use of genome editing to actually validating beneficial alleles that are present in adapted yet unproductive breeds of chicken. And most of this work I'm presenting to you today was funded through the Center of Tropical Livestock Genetic and Health by the Bill and Melinda Gates Foundation. And the idea was we're interested in looking at local unproductive chicken breeds, identifying the useful alleles in these breeds, and actually validating that they would be useful for breeding, for introducing into breeding programs. So again, I would like to say we would like to circumvent all the controversy around should we be eating or producing genome edited animals, but saying this is actually a very useful trait to understanding useful genetic traits that are existing in, are already present in chicken breeds. So to get into the heart of the matter, so I'm sure that most of the listeners are aware that genome editing of animals relies on existing reproductive technology that began 25 years ago with Dolly. Usually this entails the introduction of the isolation of an early zygote from a mammalian embryo, which can be cultured in vitro, and the injection of your genome editing tools into the zygote. This can then be reimplanted into a surrogate host animal or mother, and then she will bear this zygote to birth, and then hopefully if you injected your tools properly, the animal would be genome edited. So this has all been very well developed in mammalian species. The problem with birds is that both cloning and genome editing has been very challenging, and that's due to the avian physiology. So here what I'm showing you is an egg or an oocyte immediately after it was ovulated, and it's traveling down the oviduct, and it will be laid in about 24 hours. And this huge yolk-like structure with the egg white is a single cell, and this is the oocyte. And on top of this oocyte is a single pool of cytoplasm containing the nuclei. So if you wanted to genome edit this animal, you would have to kill the mother, isolate this oocyte, and inject your genome editing tools. This is very difficult. You could try to do this in the laid egg. So after the egg is laid, you could try to inject your tools. Unfortunately, the laid chicken egg contains 60,000 cells. So it would be very difficult to edit all these cells. And also you could perhaps try to introduce your tools into the spermatozoa, which is possible, but also very difficult for mammalian species. I want to also point out that cryopreservation of avian species is also difficult for the same reason. The large size of this egg makes it very difficult to cryopreserve, and also we can't cryopreserve the chicken egg. Okay. So rather than trying to edit this oocyte, we've actually switched to using avian stem cells for genome editing and biobanking. And here I'm showing you a video of these cells in culture. They're highly dynamic and they're highly motile. And these cells happen to be GFP positive here. So we can see their fluorescence. You can see they have these beautiful projections from the cell surface. So these cells that we're using for genome editing are actually the self-renewing stem cells that make the sperm or egg. And these are called primordial germ cells. Primordial means young. These are the young germ cells of the embryo. So this is before they've undergone meiosis to form the gametes. So these cells are diploid. I should say, these cells are very difficult to culture. You've probably heard we're trying to culture them from mammalian agricultural species and also for human, but they're very difficult to culture in vitro, except we're actually able to culture them for one bird species very efficiently, and this is the chicken part. This is the chicken. Okay, to give you a little bit of background, the chicken primordial germ cells are born very early in the embryo. And this is different than mammalian species. So in mammalian species, the germ cells that make sperm or eggs are born during gastrulation. So when the embryo is gastrulating, these special cells are set aside. Well, for bird species, we think they're already there when the egg is first fertilized, that there's cytoplasmic determinants that are segregated into the germ cells, and these specify these special germ cell lineages. And in the laid egg, so I'm showing you a schematic here of the laid egg up through the early stages of gastrulation, we think there are 50 of these primordial germ cells already present in the avian egg. These migrate through the embryo. For some reason, in all animal species, these cells are highly migratory. They're born in one location and they migrate through the embryo on their journey to the gonad. So in chicken, they start to migrate out of the embryo. Later on, they move into the blood system and they migrate through the blood system on their way to the gonad. There they form the stem cell population that make the gametes. In males, these are the spermatogonial stem cells that will form the male spermatozoa. In females, these will form the oocytes. So they replicate in the embryo, just like in humans, and then they undergo terminal meiosis, and they will no longer replicate, and they make the oocytes that will last to the length of the female life. Okay, so this is showing a picture of these cells. These are, again, the primordial germ cells. These are the precursors to the sperm or eggs, and we're able to grow them from one bird species, and this is the chicken. And this is showing these cells in culture here. How we do this is, well, it's very nice. We don't have to do surgery because the chicken is in a laid egg. We're able to take the egg and cut a little window in the shell, and that shows us the embryo. And as I said, these primordial germ cells are migrating through the blood of this embryo. So we can simply take these cells, isolate a microliter of blood, and put them in culture, and these cells will proliferate for many, actually indefinitely. So they're a stem cell population, and they will proliferate up to, starting from 100 cells, they will proliferate up to several 100,000 to 1 million cells. And my lab, several years, tried to figure out how to grow these without serum, because it was very difficult culture conditions for growing these, similar to mouse embryonic stem cells. And we came up with a cell-free defined medium to be able to grow these cells. And I should say, we've transferred this to other laboratories, and there's many laboratories now that are able to culture chicken primordial germ cells. So in this way, we can take these cells and we can simply cryopreserve them or biobank them. So we think this is one way that we can actually conserve bird species or reproductive cells. Sorry?
No problem. Mike, you continue.
Okay, thank you. Sorry. So this is one way that we can biobank different bird species using these primordial germ cells. At a later point, and this is just a movie showing this process. So again, this is that the chicken hatches at day 21 of incubation, and this is a day 3 chicken embryo. And we simply cut a window in the shell, and we go through with a simple glass Pasteur pipette, and we sample a microliter of blood. So I should say this has very low welfare impact. The chicken embryo at this stage has not developed a nervous system. It has, this is the whole body here, if you can pick it out here, and this is the beating heart. So the heart forms very early, but the nervous system hasn't formed. And we can also, you can reseal this egg and you can incubate it for hatching. And that's that's the microliter of blood we take out, which contains about 10 of these primordial germ cells. Okay, so we can use these cells and we can then introduce our genome editing tools in these cells to edit them in vitro. So we can transfect them with these CRISPR-Cas9 vectors that we heard about from Dr. Wang, and we can clonally isolate them, and we can analyze them before we make a chicken. And again, what we do then is we introduce them back into an embryo, and this is our surrogate host embryo. They will migrate to the gonad. They will go ahead and form functional gametes in that host. And this is showing, in this one case, we introduced a GFP transgene into the primordial germ cells, and we can show that some of the offspring here contain this genetic modification of the primordial germ cell. So this is the technique we use to produce our edits. So for birds, this is a really efficient system, but it does have some caveats. So what we do is, again, to summarize, we isolate the blood from an early chicken embryo. We put these cells into culture where we can genome edit them. We microinject them back into a surrogate host. We reseal the egg then, no surgery is involved. The surrogate host grows up to an adult. We can then, if it's a male, we breed it to wild type female, and we produce some heterozygous gene edited offspring 6 months later. So this is fine. This is a very nice system, but in order to look at gene function, it will take us 6 months to produce heterozygous. They have to reach sexual maturity, and it will take us a year to actually make homozygous offspring. So it's a very, for us, we've considered a slow and laborious process. And this is just showing one of our first publications, and this seems a lifetime ago, but it was only in 2017, where we used TALEN vectors, not CRISPR-Cas9, to genetically modify chicken primordial germ cells. So let me just check the time. Good. In the next 15 minutes, I'd like to tell you how we've taken this basic system and improved it to make it more efficient. So what we realized, and other people working in mammalian species realized, is that, well, if the endogenous germ cells are not present in our surrogate host, we will get much higher germline transmission. And if we could engineer a surrogate host that actually lacks endogenous germ cells, we will get 100% germline transmission. And we came up with this idea, which we call surrogate sire, surrogate dam, surrogate host mating, in which we inject our gene edited primordial germ cells into sterile surrogate hosts. And we do this for both sterile males and sterile females. And when we breed these animals together, we will produce pure offspring that are 100% from the donor cells and also have homozygous genome edits. So to do this, I'm sorry, I'm having trouble here. I cannot. Okay, I can't hide this. The Zoom sorry, the slide viewer is at the top. To do this, we produced our first plan. Well, our first plan was to make a simple knockout chicken, but our second plan was to produce inducedly sterile surrogate host. So to do that, we genome edited a transgene into a genetic locus that is only expressed in the germ cells of the animal. And the gene we targeted is called DAZL, and DAZL is known to be expressed only in the germ cells of actually mammals and bird species. So the DAZL protein, messaging protein, is only present in the germ cells. And what we did is we used a CRISPR-Cas9 vector to target the final locus of the DAZL gene, and we recombined a gene that's called inducible caspase-9 and a GFP reporter in the genome at this locus of the chicken Cas9 gene. And this is showing it here. And what this inducible caspase-9 gene is, it's actually a caspase-9 reporter gene or caspase-9 transgene that is fused to a dimerization domain. So caspase-9, you might have heard of this gene, is involved in the apoptotic pathway. And when caspase-9 is activated in a normal cell, it leads to its dimerization and leads to apoptosis in that gene. So the inducible caspase-9 gene actually contains an inducible dimerization domain called the fork-head binding protein. And this is fused to the icaspase-9. So when this compound transgene protein is produced, if a chemical compound is introduced into the cell that is recognized by the dimerization domain, it leads to dimerization of the fork-head binding domain, which causes activation of the icaspase-9 gene. And since this is only expressed in germ cells, this leads to induced apoptosis of the germ cells. So to prove this works, we produced this line of genome-edited chickens containing this transgene that's in the DAZL locus. And then we took embryos from that and we added the dimerization domain. So I said these chickens express the icaspase-9, but they also express a GFP reporter. And here we're looking at the developing female gonads of an embryo. And here are the primordial germ cells that are showing this GFP expression here. And in this embryo, we added the dimerization drug. And you can see that the gonads have developed normally, but there's no GFP expression. So all the germ cells have apoptose. And we recently described this icaspase-9 chicken in our publication in Nature Communication that came out in January. So the premise behind our new system called sire-dam sterile surrogate host mating, again, is we isolate embryonic blood from a chicken embryo, and we isolate it from male embryos, and we isolate it from female embryos. And we carry out our genome editing in the primordial germ cells from both a male and a female embryo. We inject these separately back into our icaspase-9 surrogate host with the compound that leads to activation of the icaspase-9 gene. These surrogate hosts hatch, containing only the edited germ cells, and then these can be mated together. And 6 months later, we've produced pure edited offspring containing our genome edits. So as a proof of concept, we decided to edit a genetic locus that's important for feather color. As you know, people have been studying, chickens of different breeds have a variety of feather colors, and they've been selected by breeders for many years for having different colored feathers. The gene we chose to edit is actually a naturally occurring mutation, and this is called dominant white, and this is used very often in the commercial poultry breeding because it gives a white color to the feathers. So it leads to a bird that it leads to a bird where that's when it's produced, you don't have to singe off the barbs to the feathers for it to be used for consumption. So it saves, actually it speeds up the processing of chicken for commercial breeders. But anyways, this mutation is a naturally occurring mutation that gives white feathers here. And work and this was probably by 15 years ago by the lab of Leif Erikson, identified the dominant white mutation. And simply what it is a 3 base pair insert into the PMEL17 gene. And the PMEL17 gene is important for melanocytes to deposit feather color into the chicken feathers. And when this gene has this 3 amino acid insert, it no longer can produce the feather colored or pigmented feathers. So what we did is, this is showing quite a bit of work, but done quickly, is that we could use HDR directed repair with CRISPR-Cas9 to remove this WAP insert in male and female primordial germ cells. We then injected these back into our surrogate host chicken, and we directly bred the surrogate host together. So on the left here, we are showing you wild type chicken. So this is a breed of chicken that contain this dominant white allele, and they have white feather color. And these are chicks, and these are the adults that grow up from them. And I should mention, we use a breed of chicken that have this floppy comb here. So it is a particular breed of dominant white chicken. And on the right, these are our offspring from the surrogate host. And you can see the chicks, rather than having white feathers, they now have these black feathers here. And when they grew up, we were expecting them to actually have black feather color, but it turns out in this breed of chicken, hiding under the dominant white is a different allele or a different mutation that they actually contain a gene that makes barred feather. So when we remove the dominant white allele from these chickens, the adults had, the chicks had black feathers, and the adults had this barred feather color, coming in 2 different flavors or 2 different varieties in the females, and the males have these white feathers and these black spots. And this is actually normal for a barred feather chicken. So in the final, let me check my time. So I think I have 10 minutes left still to present to you. So I would like to say this first part was how we have used genome editing to investigate gene function. So we can edit a gene in PGCs and then produce offspring from any breed of chicken to investigate the function of this. But what my lab is really interested in is actually using this surrogate host technology for biobanking. And as I mentioned at the beginning, that it's actually very difficult or impossible to cryopreserve different breeds of chicken because we can't freeze down eggs. We have no way to preserve the whole animal. And we can freeze down semen from different breeds of chicken, but since we don't have frozen eggs, we can't actually use it, and we can't bring back a breed of chicken from frozen material. So the idea is that, as I said, we can easily culture primordial germ cells from different breeds of chicken and cryopreserve these. So the idea was we could use this technology to freeze down germ cells from different breeds of chicken and then put them back into our surrogate host, breed those to reproduce a breed of chicken from cultured germ cells using this same platform. And this is the slide to show you this technology actually works. We culture primordial germ cells from a breed of chicken that has black skin. This is a silky breed of chicken. And we injected this back into surrogate host. So these are our surrogate hosts. These are our ICAS-based surrogate hosts again on the left here, and they have white skin. And this is showing the offspring here, and they all are pure silky offspring, and they all have black skin. So this is showing the power of this technology. And I've been working with researchers through the CTLGH who work at ILRI in Nairobi, Kenya, and they've adopted this technology to try to cryopreserve local or indigenous ecotypes or breeds of chicken in Africa. And they've cultured primordial germ cells from 7 different ecotypes in I was going to say, I'm getting my flags wrong, in 3 different countries in Africa. And sorry, 7 ecotypes, 5. So sorry, so this is from, let me add this up. This is from 15 different ecotypes of chicken, and they've produced over 466 genotypes that are now in the freezer. And we think we can demonstrate bringing these breeds back, we can reconstitute these breeds using our surrogate host. So in the last 5 minutes, I'd like to show you some unpublished data and how we're actually trying to improve this biobanking system. So as I said, we can culture primordial germ cells from different breeds of chicken, put them in culture, expand them in culture, and freeze them down. At a later point, we can put them back into our surrogate host chicken. The problem when we do this is that it's very labor intensive, and also that when we put them back into the surrogate host, we'll be injecting a very few genotypes back into the surrogate host. In this case here, the chicken that we produced here, we injected 2 males and 2 female lines. So all these chickens are actually full and half siblings to each other. So for poultry, it's very important that we conserve biodiversity, because poultry breeds, on inbreeding, they lose fertility very rapidly. So we have to actually be able to capture multiple genotypes. And the idea was, as I showed you before, is that we culture germ cells because there's very few of these primordial germ cells in the embryo. But as I showed you, when the embryo gets older at embryonic stage, there's over 10,000 of these cells that are still present in the embryo. And this should be sufficient for biobanking purposes. So we think we can biobank this reproductive material and cryopreserve it and eliminate the culture step. And as a proof of principle, we did this using embryonic gonads from red and green embryos, injected it back, cryopreserved the gonads from these directly, and froze that. We later injected this material back into our surrogate host, coming from both red and green embryos, and bred from that. And we showed this is actually true. We produce pure offspring from this red and green donor material directly in 1 generation. So we think this is actually going to be a very powerful tool to biobank different breeds of chicken. Number 1, we don't have to culture the germ cells. The material can be cryopreserved directly. And again, we use our sterile surrogate host to recreate different breeds of chicken. And I'll just summarize here. So today I haven't shown all the data. We've been able to genome edit 2 different breeds of chicken and validate 2 different feather traits. We think this ICAS-based 9 sterile surrogate host is very useful. It adheres to the principles of 3 Rs, as in using a welfare system to be able to biobank and reproduce chicken. And we think hopefully going forward, this will be a very useful conservation tool for cryopreserving different chicken breeds and lines of chicken. And one final question I'm going to leave you with before I take questions is that, well, are we going to be able to apply this system to other bird species? And I'll say so far, we haven't been able to propagate the primordial germ cells from any other breed of chicken besides the any breed of chicken besides the chicken. So without being able to culture these primordial germ cells, we really can't investigate. We can't really investigate this technology and how to go forward. But I'm hoping that some other lab, hopefully in India or somewhere else, will be able to culture primordial germ cells from any other bird species besides chicken and take this technology forward. So just a quick slide to help thank my funders, and quick slide to show all the people in my laboratory and my colleagues in other institutes who help with this work. And I'll stop my presentation right now. Thank you.
Thank you, Mike, for the excellent presentation and sharing of the data. And I agree with you that the working with chicken, particularly primordial germ cells, and to optimize the genome editing technique in these birds, particularly in chicken, is really challenging. But you have good success in some of the applications. You have produced these genome edited the chickens by transferring or delivering of the gene editing tools in the primordial germ cells, and then back into these embryos, early embryos, and then the live birds. Thank you. I think we have a couple of questions in the Q&A box. The first question from, again, the Dr. Basudev Kushwaha. Could you explain the technique in brief, how to separate the primordial germ cells from embryos and testes?
So for the first technique, it's really nice. So when we sample the blood, there's probably in a microliter of blood, there's 10 primordial germ cells, and there's over probably 10 million blood cells. So the blood cells all lyse in the first week of culture. So the primordial germ cells lyse, and the blood cells lyse, and the primordial germ cells will then proliferate in culture. So and I should also mention that they grow in suspension. So we simply isolate the suspension cells. For the gonad, we can do the same thing. We can take the embryonic gonad, you can dissociate the cells from the gonad in culture, and the primordial germ cells in the gonad will still proliferate and grow in suspension. So we can enrich that way. In terms of biobanking, for the male, we don't have to isolate the germ cells from the rest of the somatic cells. We can simply take the gonad, dissociate the cells, freeze them, inject them back into an early embryo, and the germ cells remigrate and populate the gonad. So that's for the male. For the female, what we do is we use magnetic cell sorting to an antibody called SSEA1, which is expressed by the gonadal germ cells to enrich. So in the female, we do enrich for the female germ cells from the frozen material and inject it back.
Yeah,
I hope that answered the question.
Yes, thank you. One more question from this same Basudev: how to produce the inducible sterile surrogate host? How to produce inducible sterile surrogate hosts?
Yeah, so what we used a system or a transgene. So I should say that's the problem also. The host that we're using isn't genome edited. We use genome editing tools to produce it, but we used an iCaspase-9 and also a GFP transgene that we targeted to the Dazzle locus. So we're able to modify any genetic locus we would like of the chicken using primordial germ cells. So we use homology-driven recombination to insert the iCaspase-9 transgene into the Dazzle locus. So all the chicken contain it. It's not active until we give the prodrug. So the iCaspase-9 protein is present in the germ cells, but it's inactive only till we add the dimerization domain. So the system has been used in mice very often, and also it's used in human genome therapy also to ablate cells that aren't needed.
Yeah, thank you. I think there are a couple of questions, but because we are running a short of time, so I will take 3 or 4 questions that is important. Maybe I will share these questions to you, Mike, and you respond, and then I will share to all the attendees. We have more than 190 attendees, so they can also understand if they have. There's a few questions. One question from Sujoy K. Dhara. To isolate the PGCs, does the sex of the embryo matter? Here are a couple of questions. First question is, does matter? Second question, for transferring of PGCs, do we need to transfer back them in a ven or anywhere in the developing embryos? Other, for culturing, he is asking, I think, I don't know, you will able to answer it or not, or you may want to share or not, that the serum-free media you have discussed, so its composition is available in the public domain or it's commercially available?
Oh, no, that's what we published. We've published, and I have detailed protocols I can give any laboratory on how we culture the germ cells. The medium is commercially available. Unfortunately, we use, so I should explain this, the cells, the primordial germ cells grow in suspension. So we developed a medium that was low on calcium. So we had, I should say, Gibco produce this calcium low medium. So it's very expensive for us to buy, but there are some commercial alternatives that are present. Yes. And the first question, sorry. So you can introduce the germ cells back into the vascular system. So when I showed the sampling of the blood, you inject the germ cells back at the same stage, or you can actually put them back into the laid egg. You can inject them under the forming epiblast in the embryo. When the egg is laid, you can put your germ cells back at that stage, and you inject several thousand, and they'll remigrate back to the gonad. So they have to be introduced at these very early stages of development.
Thank you. 2 more quick questions. You quickly respond. One from the Sunil Kabgate, another from Vikrant Singh. How the gene editing help to develop the diseases in chicken breeds development? Is there any development happening in terms of the disease-resistant chicken breeds? Second question from Vikrant Singh is how to separate gene-edited cells? Because in the PGCs, this may be from my side also, because you have the millions of cells, you are transfecting them. So do you separate them before transferring back into these into the ven?
Yes. So we I'll go to the second. You have to remind me of the first question, but
yes.
The second question is you can cell sort single cells, the primordial germ cells. So you can transfect for genome editing. You can take a single cell, put it in a well, grow it up, analyze it, see that it has the edit you're interested in, and then freeze that down, and then at a later time introduce it into the surrogate host. So that's why we grow the cells, we edit the cells clonally. So that's how we determine they have the edit we need. And what was the first part of the question? Sorry.
How the genome editing help to develop the diseases in chicken? Any improvement is there?
So so many laboratories are working on that. So the idea will be we can validate genes that are important for disease resistance, and we could, as I say, they could be used to introduce into breeding programs later on, or perhaps if genome editing is accepted, the animal could be used. So we have the very important genes for us in terms of poultry disease are the ones that are going to be the most difficult to edit. This is Newcastle disease virus. This is avian influenza. And this is also probably for Merrick's disease and Eimeria. And none of these really have a receptor that we could edit to keep the virus from getting into cells. So we do have one project that was out there in the media that was released, that we have a gene that's called ANP32A that's important for replication of avian influenza in chicken. So we're investigating that to see if that gene is edited, could it lead to increased disease resistance? But that's our problem. We can edit, but we don't have that very many targets, to edit.
Yeah, thank you. The one technical question, last questions from Dr. Abhishek Bharadwaj. He's asking what should be the diameter of the glass pipette for efficient collection of PGC's when
we use, well, no, I don't want to say that on a live thing, is that we use the standard tools that is used for human embryology and also for your work in Buffalo, where you pull glass capillaries, you break the ends of them or you file the ends, and you use them for transfer. So you can simply do this by hand. You have a 1 millimeter glass capillary, you pull it on a flame, you break the end, and then you use that using aspiration techniques. You go in and you can sample the blood that way.
Yeah, so it's usually for the embryos of 400 to 500 micron is
exactly. Yeah, I should say for that. I should say 50 microns to 100 microns in size.
Yeah.
That's a short answer. Thank you.
Thank you, Mike, for your excellent presentation and the answers. Now, we're moving ahead. Before we go to the closing section, now I invite the Honorable Joint Director Research, Dr. Dheer Singh, for his remarks. Hello. Hello, Dheer Singh. Hello. Okay, maybe he's busy in some other assignment. So next time, invite the Dr. RRB Singh, who is the head of these or in charge of the NAHET project, which aim was to bringing up these or excellency or to educating the students as well as faculty on the higher education, agriculture, particularly agriculture education, as well as his joint director academics. So now I invite the Dr. RRB Singh sir for his remarks. सर अनम्यूट कर लीजिए
सर अनम्यूट कर लीजिए
सर अनम्यूट सर आपका
am I audible now?
Yeah,
okay.
yeah.
Yes, yes.
Okay. So at the outset, I thank both Professor Wang and Professor Mike for having very kindly agreed to be a part of this exercise. And in fact, tomorrow also we have 3 presentations. We look forward to those presentations as well. And in fact, see, I am not a man of biotechnology. My area of expertise is something different. But what I would like to share with you is that this entire exercise of bringing scholars from around the world to deliberate and to interact with our faculty and our students is being done under one program which we are running for last couple of years, and that is institution development program. In fact, in this particular program, we have a provision of inviting professors and also sending our scientists and students to overseas laboratory to see for themselves and to interact and to come to know as to where the science is progressing, what kind of work is being done in different laboratories across the world, so that our faculty and scientists, they become aware of it and they are able to establish network. And that is one of the major what you call tangible outcomes that we look forward to from this entire exercise. So I think because of this COVID situation that prevails, for our scientists to physically go and work in those laboratories and also invite faculty from overseas to be a part of the faculty for maybe a week or maybe a couple of weeks at our place. So this has not been possible. So we thought that probably this could be a better way of at least continuing with our exercise that we started some 2-3 years back. But certainly whenever there is an opportunity,
I think some connection issue is there.
To our campus. So that is one thing that we will sincerely look forward to. And at the end, I can at this stage only say that your talk have been very fruitful and very useful for our faculty. And we hope that this kind of collaborations or rather interaction continues in the times to come, so that whatever we look forward to gets strengthened. So I thank both of you for being a part of this entire proceeding. Thank you, sir. Thank you, both of you.
Thank you, sir, and thank you for your motivation and encouragement. And I hope when the COVID situation will be subside or there is no COVID cases, we will have more even in presence and we will have the chance to invite the eminent faculties from the overseas. Thank you, sir, for your leadership and your motivation. Now I invite the head of the department, Dr. Sachinand Day, for his remarks.
Okay, this is a great talk. I think many people are benefited out of this particular, both the lecture and both the speaker have described very nicely their ultimate outcome as well as basic principles of that. Throughout the presentation, I have seen almost 180 to 190 participants are there. So this is possible because of this online mode. If it is in the offline mode, I'm sure this much of participants cannot be benefited out of this e-lecture. I think thanks, Mike, nicely described this all these difficulties in this poultry. Although the generation interval is very small in case of poultry, but it is very difficult to handle to use this particular technology. And your one presentation, this is not only this is a CRISPR genome editing, it is a breeding and reproductive technique. So until unless both these technology merge together, it is very difficult to get the benefit of that. And whatever genes you have targeted, they are all single gene. They are going to give the color. That is whether it is skin color, whether it is a shank color. Also, they are also in one of your slide. You have shown there was a black color shank and yellow color and white color. So that is also a single color, single gene controlling that particular locus. It's a great talk from poultry. I think many people poultry they may not be knowing how difficult they handle this thing. And regarding this Wang presentation, I found this is very nice. Although this CRISPR-Cas started popularizing in 2012, but they picked up very quickly, and in 16 they started publishing this particular work. And they have a lot of work in sheep and goat. And I think both the talks are beneficial for us, those who are working in mammalian system. Although in mammalian system it is a little easy to handle and to process also. Compared to poultry, no doubt. Poultry is no doubt a difficult part, whatever you have shown. Okay, thank you all for this nice presentation, for selection and for providing your time to share your knowledge. And even if you some unpublished work also you have shared. Thank you, Mike, and thank you, Wang.
Thank you.
Thank you, sir. So Joint Director Research has joined. So I invite the Honorable Joint Director Research, Dr. Dev Singh sir, for his remarks.
Thanks, Naresh. Unfortunately, it was very important topic, so I only listened actually the one lecture initially. But I think no need to be say this is a really very exciting technology and techniques, and we need to be improved. I think you are already going on, so you maybe get benefited from all these techniques, and we need. I think we should have not only if you can make the buffalo and change the genetic, but we can go for the functional validation of genetic function. Because I think at this moment, if we can make the functional validation of a particular gene into using this technology, that's also be a very great help. Because later on, once you identify that this gene has this role, then the manipulation is not very difficult as far as the embryo technology is concerned or IVF. So major answer, I think that's a lecture and this collaboration with this all the three speakers were really excellent. And I have also experience about gene functional validation of gene using siRNA and all. But now the CRISPR is come as a gold standard, I think, because not only the functional, but it can be implemented into the large animal for the application also, because it's considered as a non-transgenic. That's more important. So thank you very much. Once again, I also thanks actually all the speaker for excellent talk. And thank you, Naresh. Thank you very much.
Thank you, sir. We will continue the efforts in the genome editing areas, particularly for dairy animals. Before closing, once again, thanks to the Dr. Mike and the Dr. Wang for accepting our invitation and deliver the talks. We have the two more